Hello, everyone. Welcome to our sixth and final SERC Talk of 2022. This talk will conclude our Innovating for STEM Readiness series. And thank you so much for taking the time out to join us today. For those joining for the first time, I'm Mimi Marcus, the SERC Talks webinar coordinator. We hope you find this discussion beneficial and encourage your active participation. So as COVID has provided us a familiarity with all the participation tools, I'll just remind you that you can utilize the chat for comments. Feel free to let us know where you're joining from. But if you would reserve the Q&A module and for questions that you'd like to pose to our speaker and moderator, but feel free to use the chat and reactions buttons as you see fit throughout the discussion. While you are muted, you can utilize those different aspects. If you are joining by phone only, please feel free to send your comments, questions to serctalks@stevens.edu. And again, you'll be able to find the slides and the recording of this event following at uark.org/SERC, SERC-talks, plural TALKS. With that said, it's my pleasure to welcome SERC friend and Research Council member, Dr. William Rouse, as our Innovating for STEM Readiness series moderator. We're very lucky to have Dr. Rouse, who in addition to his Research Council leadership and research roles within the SERC, and Eric, the Acquisition and Innovation Research Center, is a senior fellow in the office of the Senior Vice President for Research at the McCourt School of Public Policy at Georgetown University. Dr. Rouse is also our most recent recipient of the SERC Founders Award, our highest honor of our UARC. And we thank you again, Bill, for curating and moderating this series. So with that, I turn it over to you.
Well, thank you very much, Mimi. This is a great honor to be able to introduce Dr. Rita Colwell for this talk. She's done some amazing work for a very long time. I was fortunate enough to serve on a academy committee with her and got to know her a little bit that way. But the bio sketch that's put online is just really amazing. I've never had a chance to introduce anyone who has had a mountain named after them in Antarctica in this case. She's a distinguished faculty member at University of Maryland and Johns Hopkins, and has founded a really innovative company, CosmosID. If you go into their website, it's really, really pretty cool. Rita's going to present a case study, if you will, of a highly interdisciplinary study with profound impact. And we're going to use that as a basis for later discussion of how does education prepare people to do those kinds of studies that are most ambitious and really impactful. And I won't take any more of Rita's time. Just welcome to the SERC Talks, Rita, and I'm looking forward to hearing your presentation.
Well, thank you so much, Bill. It's a great honor and pleasure to join you all and have the opportunity to talk about my favorite subject, which is cholera and pandemics. The important point I think to make is that, see if I can get this to advance. Not advancing. There we go. We've been in two pandemics. Obviously, we know the COVID very well. It's been a disaster in so many ways. But starting in the 1960s, the seventh pandemic of cholera has been underway and has been affecting particularly the lesser developed regions of the world. Hence, it's sort of out of sight, out of mind. But it is a pandemic. It affects, if you measure just diarrheal diseases, which includes cholera, it's about 1.5 billion cases every year and close to 2 million deaths. So this is an ongoing pandemic. It's a global disease. Cholera is a water-related diarrheal disease, and as I just pointed out, the seventh pandemic, the waves of cholera epidemics started in the 1960s, and it's affecting about 50 countries and about 7 million people. Unfairly, the Bengal Delta, the region just offshore, or I guess the coastal region of Bangladesh, is where the cholera epidemics tend to be most severe. And they've been known as the native homeland of cholera. But the point is, as I'll point out, as I'll show in my study presentation today, the bacterium is distributed in aquatic habitats throughout the world. And that 100 or so years ago, we had cholera in the United States, in Canada and South America, and in other parts of the world, including Europe. We made the discovery that the cholera bacteria exist in the aquatic environment. And the serotype 01 has been the epidemic strain. But we've learned that we can't eradicate cholera because the bacteria are in streams, coastal waters, transported in oceans throughout the world. The disease is straightforward in that the bacterium is readily cultured. The bacterium in the upper right is a rod-shaped bacterium, polyflagellum, easily cultured on a medium that has a dye that can allow selection for the colonies in the sample. And the disease itself is highly enervating. It's a loss of fluid that can be so severe that systemic shock ensues and then death follows. The discovery we made in the early 60s, or I and my team, and these numbers are the stations that we sampled throughout the Chesapeake Bay, was that the bacterium is in the environment. I can tell you that that was not well accepted. I was challenged every point of the way to the present, but now it's in textbooks. We discovered that the bacterium, which all Vibrios, of which Vibrio cholerae, causative agent of cholera, is a member, carry an enzyme, chitinase. It breaks down the polymer N-acetylglucosamine, which comprises the shell of crabs and shrimp and copepods. And so the bacterium, the Vibrios, recycle the crustacean remnants and also participate in the carbon and nitrogen cycle, which makes them really vital to the environment. The Vibrios in the natural environment can be isolated from a variety of animals and plants in the aquatic system, but it's really the copepods, which comprise about 90% of the zooplankton populations of the Earth's oceans, therefore the predominant carrier or host, if you will. I prefer to call it the vector. It doesn't fly like a mosquito for malaria. It swims, but it carries the Vibrio cholerae. And so we very early on developed a model way back in the late 60s, which was disputed constantly by the medical community. They believed fervently that cholera was transmitted person to person. But the environmental factors that are in a system, like our environment that we live in, are critical to our health. And I think only recently with COVID have we begun to understand that the immediate environment is very important for human health, and it cannot be ignored without trouble and travesty. So we developed this model of the copepod, shown here as the vector. And this work was done in the Chesapeake Bay. And the medical community said, well, that's just one of Colwell's crazy ideas that works in the US, but does it work in Bangladesh? That was a challenge that I took up and went to Bangladesh with my students in 1975. That was my first trip to Bangladesh. And sure enough, we showed that, yes, person-to-person transmission is critical when you have an environment with poor sanitation and without the medical capabilities, you have a person-to-person transmission. But fundamentally, the origin of the bacteria are the immediate environment. So we were able then, in even more recent studies, we've been able to develop drug-resistant profiles of the Vibrio cholerae from the work that we did in over a 14-year period funded by the NIH. In this case, it was a collaboration through my connections with Johns Hopkins and the University of Maryland. And we were able to show that changes over the years, over the last 4 or 5 years, we can see in the kinds of antibiotic resistance factors. What really struck me in about 1985, Landsat satellite was launched. It measured chlorophyll, the ABC components of chlorophyll, sea surface height, and sea surface temperature. Those were factors, including salinity, that drive cholera epidemic based on all of the work we had done in the Bay of Bengal along the Sundarbans, where cholera is endemic year after year. And so it occurred to me that we could use satellite imagery, sensing, to be able to predict the risk of cholera. And sure enough, what we did was an analysis. We published this 22 years ago in the Proceedings of the National Academy of Science, showing the blue line is the satellite measurement in a, and this was very crude, it was a 1 mile quadrant off the Bay of Bengal. And then we measured the summation of cases in red in the villages along the Bay of Bengal. And the correlation was really astounding, considering we're measuring satellite sensing parameters of the water and counting the numbers of patients succumbing to cholera in Bangladesh. And so that really provided us with a dramatic capability to further show that cholera occurs in 3 different forms. Epidemic, which is in the more remote regions of Bangladesh, where there's a concatenation of weather parameters and a human movement for a religious festival, an accumulation, a crowding with poor sanitation. And then you have this epidemic and there may be years between the big, large number of cases. But we also were able to show that along the coast, Mathbaria being typical, and Bakaganj, cholera occurs all year long, with large numbers in the spring and fall, which correlate with the blooms in the plankton. And then Dhaka city has a mixed mode, usually a couple of spring, fall peaks, but this can be modulated. And if you have a severe weather event, then you have an epidemic. So the environment plays a very significant role. We've since made much more sophisticated the model, and we've focused predominantly on epidemic mode of cholera. This takes into account rainfall, warm temperatures. If there are very, very hot seasonal temperatures for 4 weeks, followed by a very heavy rainfall in an area with poor sanitation and lack of access to safe drinking water, the risk then will be extremely high. We now use a variety of satellites, very sophisticated. We started out, of course, with the Landsat in 1985, 1990, moved on to more sophisticated forms of Landsat, and now we're including a variety of the what were classified satellite sensing capabilities that allow monitoring movement of populations that are now available for scientific research. And we've been able to improve our prediction from about 60% accuracy to about 90% or more. And we tested it in 2017 when there was the massive cholera epidemic in Yemen. What we did was calculate and compare our prediction using our model, which is shown on the left in multicolor, with the actual cases shown in the lower right. And this then has been picked up by UNICEF and the UN agencies, because what it allows is being able to show the risk location where it would be highest. And then medical supplies and safe water units, et cetera, can be provided in a very effective, efficient way. And in fact, we did that in 2018, and were able to reduce cholera significantly because we were able to mark by monthly maps provided to the UN agency the high risk areas where they could emphasize their work to prevent and to deal with cholera. I'm going to shift now to the other aspect of the work that we're doing, which started when I started at CosmosID back in 2007, where I simply, because I formed a company because I figured the $2 million that I needed to get this launched, I would not be able to get in one lump sum from NIH. So starting a company seemed to be one way of getting the science going. And that is, we've developed the capability, which now many others do, but this was 15 years ago when the idea and the work began, where we can extract DNA and RNA, and the community DNA can be sequenced, and the sequence reads are matched against a database that we've established over now close to 200,000 bacteria, viruses, fungus, protists, and that allows us to do a matching. It's a very successful identification of all of the microorganisms, bacteria, viruses, fungus, and protists. And also we can analyze the genes that are present for antibiotic resistance and pathogenicity properties, and currently working on metabolomics and actually mapping the interactions of the chemical reactions, which gives us microbial identification and pathogen detection. This of course is much more effective. The Meldetop approach can give you very accurate antibiotic susceptibility, but compared to whole genome sequencing, what we can do with whole genome sequencing is all of this without having to do many different steps. And the accuracy we can achieve now is to substrain typing, which is critical for pandemic and epidemic monitoring. And so with this approach, we're able then to understand the presence of pathogens, their characteristics, the presence of antibiotic resistance, and this allows, of course, being able to preempt antibiotic treatment by knowing what antibiotic resistant organisms are present in the given sample prior to treatment. And one example, which I'm abbreviating because of time, is a study we've done in Calcutta, India, where we received a series of samples, phase 1, 2, and 3, where the Calcutta laboratory, the National Institute of Cholera and Enteric Diseases, were able to determine using classical techniques what was present in terms of cholera. And if they couldn't pick up cholera, they sent us the samples as unknown etiology. And then they also provided some healthy controls. And they did all of the classical tests for this set of organisms, and we did the DNA work. And what was very interesting was when they could detect Vibrio cholerae, we were able to do so as well. But in addition, we were able to determine the presence of other pathogens, such as Salmonella or Shigella or Campylobacter. In other words, the disease is actually polymicrobial. where they could not pick up Vibrio cholerae or detect it by their classical techniques, as I showed, with a petri dish, we were able to determine the presence of what was causing the diarrhea. And in most cases, this pink area, these were enteropathogenic and various pathogenic E. coli predominantly, along with some of the other enteric pathogens. Interestingly, in the controls, we found that they are carriers much more extensively than when we compared the human microbiome program data from NIH for the Westerners, Europeans and Americans. We do carry a bit of pathogens, but compared to the number which the Indian healthy population, which we tested, were able to carry was much greater. The blue are the interesting probiotic organisms picked up in the stool samples, the DNA extracted from stool of the healthy population in India. Because of those of you who may have traveled to India or may have Indian friends or like Indian food, yogurt and probiotics are a major staple, not so much in the US. Anyway, that's a sort of a sideline. But the other important factor we were able to learn is that these are various antibiotics, and you can see that there's a higher incidence of antibiotic resistance genes in the Indian population, including the healthy population, mainly because antibiotics are available without prescription, in fact, just from peddlers on the street. We now have got to the point with our Vibrio prediction, working collaborative with me, with Dr. Antar Jutla, a former postdoc who is now an associate professor in the College of Engineering in the University of Florida, working collaboratively, University of Maryland and University of Florida. We now are able to provide online a prediction of risk in a number of countries. We're hoping to go completely global. Now, what has all this got to do about COVID? I don't need to go into the explanation of what COVID virus can do to us. I've been talking about a bacterium, and now I'm talking about a virus. It affects all of our organs and in many ways. We determined very early in, I think, March of 2020, right when the beginning of the pandemic began, that we could, in fact, pick up the SARS-CoV-2 virus and its variants by the technique that we had developed that I had just described. Others had shown that the stool, and we now are all well aware of this, carries the virus. And interestingly, this looks complicated, but it isn't. It's pretty simple. These are 41 patients, and the red line for each patient across horizontally is when the throat swab or the nasal swab is positive, and the orange or yellowish orange line shows how long afterwards the stool sample is positive for the virus. And so it's clear that monitoring wastewater is very important in being able to monitor the risk of COVID in a community. We launched a study in 2020 in the summer, working with the Department of Environment in Maryland and through the governor's office, sampling and testing wastewater facilities and sites throughout Maryland, using the pretty standard technique of sampling wastewater and using specific components of the COVID virus as part of our detection. And this is one example. The blue line is the target gene recommended by CDC, and the orange line are the additional genes that we employed to confirm our findings starting in June of 2020. And by July, we picked up a peak. This was from Frederick, Maryland, where we've been working closely with the public health office there. And we were able to provide a preliminary warning that COVID was appearing in the community. The best case study was Mount St. Mary's University, where we carried out twice weekly sampling of wastewater at the individual dormitory sites. In other words, the effluent at the particular pipe from that dormitory. And we found 10 positive. We found that one of the dormitories was positive, and then all of the students in that dormitory were tested. 10 were positive and 9 were asymptomatic. This was very early. This was in 2021. This was over a year, almost 2 years ago. And this then became a means by which the university, Mount St. Mary's, was able to stay open and simply quarantine the one dormitory. What we did was to convert the risk calculation, the model for cholera to COVID, taking into account factors such as the importance of humidity, to dew point temperature, movement of populations, and immunity, building that into the COVID risk map. And this was a very early risk map in 2020, April and May of 2021, rather. And we now have been able to take into account temperature, warm regions, cold regions, and people, the movement of populations depending on air temperature. If you're in a warm region, you go indoor to air conditioning. If you're in a cold region and the temperature is low, you go in for inside where it's warm. So we were able to build into the model an environmental COVID risk prediction. And this shows April 2021. This is the risk map, and these are the actual cases for that period of time. And we have continued our wastewater analyses and actually even monitoring in one of our studies, antibiotic resistance presence, and being able to put together the actual cases you can see in yellow, and this is for Maryland, and the blue line represents the wastewater measurement of virus in the wastewater. The red line is the satellite computational model of COVID risk. And we're now being able to put all this together so that, for example, there was a rise in prediction. There was a concomitant increase in wastewater virus being picked up. And then there was an increase in the number of cases. So we're in the process now of perfecting this approach to be able eventually to correlate numbers of cases with the risk prediction. We've gone further to do analysis of the DNA and the RNA down to bacteria, viruses, fungus, and protists, because this is a tool. This is a public health tool that we can use for community public health. You can pick up, for example, increase in Salmonella or Shigella, or even increase in antibiotic resistance, which we have detected in one of the Eastern Shore cities in Maryland. And so this is a tool for being able to be prepared for an outbreak, not only for a virus infection as COVID, but also for bacterial and fungal infections as well. We now have online the COVID risk map, and it's accessible to anybody via the internet. And that provides us then a mechanism for being able to provide an early warning for increased risk of COVID. Let me see. Let me go. I like to conclude with the quote from John Muir, founder of the Sierra Club, who said, "When one tugs at a single thing in nature, he," and then I inserted "and she," finds it hitched to the rest of the universe. And I think that's an important message for the 21st century. Many collaborators from the International Center for Diarrheal Diseases Research in Dhaka, Bangladesh, in Calcutta, India, many students, and particularly a shout out for my current student, Kyle Brumfield, who's done so much work in the latest research. Many collaborators from around the world and many other universities. It's an interdisciplinary, international, collaborative effort. And I have published a lot of this in the book that was published 2 years ago by Simon and Schuster. It's a paperback now, showing and describing not just my journey through science as a woman, but many other women and their stories as well. And I think it provides a good point of discussion for equity, inclusion, and diversity. So with that, I will stop sharing and be happy to take any questions that you may have.
Thank you. That's absolutely fascinating and profound work. One question I have, and especially when you see the list of all the collaborators, the number of disciplines involved, I mean, ranging from biology to computer science to satellite engineering, I don't know. How did you bring all those pieces together?
This is because, as I describe in my book, my journey in science has been obstacle ridden that I've had to overcome. And as a result, I started out as an undergraduate in genetics. And I was planning as a bacteriologist, and I was interested in microbial genetics. I was intended to go to medical school, and I applied to 3. Yale was one, and I was accepted at all 3. However, in my senior year, I went on a blind date with a very handsome graduate student, and 2 months later, we were married. I didn't want him to lose the time, and we both said, it'd be a good idea if I could get a master's in a year. He would finish up his master's, a master's in physical chemistry, and then we'd go elsewhere for our Ph.D. So I went to my department chair and asked for a fellowship, and he said, "We don't waste fellowships on women." No chair would say that today. They might infer it, but they wouldn't say it. I went to my undergraduate advisor because he knew I was an A student, and he was a geneticist, fruit fly genetics. And he said, "I have an RA, a research assistantship, if you want to do your master's in fruit fly genetics." And I said, "Sure." That was fortuitous because this was 1960. This was a time when genetics was just burgeoning. So I got trained in classical fruit fly genetics, but at the time at Purdue, where I was a student, Jack and I were students, to get my degree, I had to take courses in tomato genetics, bacterial genetics, fruit fly genetics, human genetics, etc. So I really was very well grounded. Now, we applied to the University of Washington because Jack, the physical chemistry department there and the physics department were excellent, and the medical school was very good. So we applied. He was accepted. I was accepted to the medical school, but then I got another letter that said, "Oh dear, you have to have lived in the state of Washington, Oregon, Idaho, or British Columbia for a whole year, paying taxes prior to being accepted to the medical school because we want to keep our medical graduates in the Northwest. So they said, as an alternative, we'll give you a fellowship in the microbiology department. And then if you want later, you can switch to medicine. Well, I got to the microbiology department. I said, okay, that's fine. So we went to the University of Washington. And in microbiology, the first thing I did when I showed up was every new student had to be interviewed to be properly placed with a good advisor. So my advisor from Purdue had told me about Dr. Herschel Roman, who was a member of the National Academy geneticist, that I should talk to him. So when I get to the microbiology department, the microbiologist who interviewed me said, well, what do you know about the UW? And I said, well, Dr. Herschel Roman would be someone I guess I should talk to. Well, turns out the fellow I was talking to and Herschel Roman did not like each other at all. So that was not a good introduction to the micro department at the University of Washington. I then spent a year working with Dr. Roman, which didn't work out very well. A new professor had come in, but I got more microbiology, more genetics. And then a new professor showed up in oceanography and fisheries, named John Liston. His name was John Liston, who was starting a new program in marine microbiology. And he needed a technician, so I ended up being interviewed and was hired as a technician. But it was so fascinating. I helped him set up the lab and so forth that I ended up doing marine microbiology. So to do that, I had to know something about oceanography. So my degree is now in oceanography. And to go to medical school, which I could have done, but it would have been very expensive and neither Jack nor I had the money. But as graduate students, we could make it through. So I ended up then having been trained in microbiology, bacteriology, microbiology, genetics, fruit fly, and all kinds of genetics. Then with Herschel Roman, who was a yeast geneticist, I became proficient in dissecting yeast spores to do genetic studies. And then with John Liston, I learned oceanography, fisheries, and general ecology. But I gathered so much data as a graduate student that it was going to be very difficult to do the analysis. But a new computer had just been installed, the first computer, the IBM 650, which has less computation than my Apple Watch. But this was 1960. And so I wrote a computer program. I was the first to write a computer program to analyze and identify microbial data. So I developed a program for my PhD thesis to identify microorganisms in communities on marine animals and on marine plants and in seawater. And what's fascinating is by all of that twists and turns, I ended up, which today, 25 years later or more, is the hottest area in science. And that is understanding microbiome of ourselves and our planet Earth. So it's been a fascinating path and a long answer to your question, but it had to be long because the path was contorted.
Well, I'm interested in your path is just absolutely fascinating, and it's kind of hard to imagine anyone planning a curriculum like that. But what about all the other people involved? How did they come together?
I think the important thing is that today it's important to have a major. It's important to be expert or at least well-founded in biology, chemistry, physics, engineering, and within the disciplines, whatever the specialty may be. However, it's really critical that there be interdisciplinary collaborations to be able to really solve problems. Because as I think is pretty clear from the cholera and COVID results that I've presented, that you have to be conversant with satellites and satellite data management and handling, as well as fundamental biology, microbiology, and ecology, and interdisciplinary weaving of the information to be able to come up with conclusions, which means big data, and the analysis of the data in a way that allows pulling out from the hypothesis the conclusions of verifying or negating the hypothesis.
So what if someone wanted to get involved with what you've been doing of late and all over the decades, what would you recommend to those students that they do?
I think it's important to declare a major, but I think it's important to be what we were encouraged to be when I started as a student, but it became much more specialized. I think we do over-specialize our students these days, is to take the courses which I did in my path in everything from psychology and sociology, a course, you don't have to major in it, along with a course in understanding ecology. And certainly, I think all students are computer conversant these days. They are from almost birth now. So being computer literate, which was a novelty when I was a student, is now, I think, available to everybody. And so that means being able to know how to handle and analyze large volumes of data to be able to pull out the more subtle conclusions and correlations, I think, is critical.
Okay, I've got one other question, and I'm going to just tell you what it is, but put it on the shelf so we let somebody else get to say. But I'm interested in whether or not the hurdles you face as a woman are easier now from your perspective. But let's say that as the wrap up and see if we have other sort of technically oriented questions. Okay, do we have any, Mimi?
Let's see. Do you have any in the chat?
Yes, and Carl's able to unmute himself if you'd like to ask directly, Carl. Otherwise, yep. Carl, I see you're unmuted. Are you testing
Hello,
out your?
can you hear me, Mimi?
Yes.
Okay. Well, first, thank you, Dr. Colwell, and thank you for your service to our country and the world. I'm so impressed and so thankful for your work. I'm currently working in the food industry, and I wondered what your thoughts were for applying some of the work you're doing at CosmosID to understanding food safety and microbial issues in food. And if you have any advice how we at home should be testing our foods or products we could develop to do it without expensive sequencers. Thank you.
Well, that's a wonderful question, and thank you for your very kind comments, Carl. We are doing work with the FDA, and in fact, we've done a variety of studies because the approach that we've taken is really ideal for detecting the presence of pathogens and being able to enumerate them pretty effectively. And I have some, at least two, maybe three. I think I have 4 former students who are now at the FDA and have been employed, some for a number of years and some recently. So I think understanding the microbiome with respect to food safety is critical. And the University of Maryland actually has a program collaborative with the FDA. I think the juxtaposition of the laboratories, I think, is fortuitous and helpful. So food safety, I think the microbiome approach is really the future.
Great, great. Do we have some other questions here that are pending?
We do. And Frank, you're able to unmute if you'd like to ask yours directly.
Yeah, we'll give this a shot. Dr. Colwell, thank you for presenting. Very interesting. I work in the space industry with Lockheed Martin Space, and I was just wondering if you had any insight into the type of payload sensor on the Landsat that was able, how were they able to measure bacteria from the Landsat?
Oh no, they didn't measure the bacteria. I hope I didn't mislead you. They measured the parameters of chlorophyll, the various components A, B, and C, and the sensor also measured sea surface temperature and sea surface height. And so what I was doing was building a computational model that took those data into the model, and then would give me a calculation of risk for the given site that was being measured. So what I had done was the fact that the relationship with chlorophyll had been shown by our ground truth work. That allowed me to confirm the relationship with plankton, because during the year, when the plankton populations became abundant, the numbers of cases of cholera would go up in Bangladesh. And so the cholera in Bangladesh for years and years has been high numbers in the spring and the fall. And we were able to correlate that with the peaks in plankton spring and fall. So by measuring, using the satellite sensing information and incorporating that information into the computational model that we developed, we were able then to predict the risk of cholera, and the presence of the cholera bacteria were confirmed by ground truth actual measurements, just as we did with COVID by measuring wastewater and the presence of virus in wastewater. But the correlation is such that by simply measuring using the satellites, we now are providing information for Yemen and Ethiopia, Sudan, and very recently we picked up signals in Mariupol, Ukraine, in June, this past June and July, of a risk of cholera. If there had been severe weather in Mariupol, in Ukraine, they would have had an epidemic, because years ago they have had cholera there, and we were showing the risk was very high, particularly since the Russians had destroyed for Mariupol the safe water drinking plant, the purification plant, and the sewage treatment plant. So the risk was very high. So it's measuring risk rather than actually measuring the bacteria directly by satellite. But the way things are going, I wouldn't be surprised in a decade that the satellites will be doing just that. But for the moment, it's really providing us with the information for our computational model and prediction. But thank you for the question.
Yeah, no, thank you for the response. My parents are ER doctor and ER nurse retired, and I work space systems. So this has been a fascinating, all around for me. I appreciate your time.
Well, let me add something. We are involved right now, and the information will be released in the next 6 months. We've been involved with NSF funding, developing a Vibrio prediction for Chesapeake Bay, and also in Florida, because what's happening is that it isn't cholera that's so much a problem. The bacteria are present in Chesapeake Bay, but we all drink treated water. But what is a problem is Vibrio vulnificus, which, if you're swimming in the bay and you have an open cut or lesion and it gets infected, the fatality rate is about 50%. And so it's serious. You really need to know if the Vibrios are abundant. So we're in the process now of developing a Vibrio index for various parts of Chesapeake Bay so that you can be alerted when you are in recreational, whether it's boating or swimming or whatever, in the bay, in parts of the bay where the risk is highest. And so I think this can be very beneficial, particularly in the mid bay where there tends to be a higher number of cases. It's not epidemic, but there are maybe 1 or 10 or 15 cases a year, but they can be very severe. There's when I give a lecture on Vibrios in Chesapeake Bay at Patuxent, there's always in the audience 2 or 3 people who have either lost a leg or a finger or even an arm from an infection that wasn't caught in time, and amputation had to be done, but their life was saved. So we're developing the Vibrio index, and I'm just mentioning that since your parents are physicians, they may want to avail themselves of the information. Thank you.
Very
Thank you.
good. Mimi, do we have any others?
I have a ton personally, but I think we've gone through the ones in chat.
Okay. Well, why don't you ask one question, then I'll get to my wrap-up question. Okay.
Okay. Yeah. Just following up on that, you said specifically in the Chesapeake Bay area, but are the Vibrios kind of on a national scale on a similar level?
Yes. In fact, we're working right now with Dr. Jula and Florida because they're having severe problems with algal blooms, and there seems to be some relationship, which we're now teasing out, with the presence of Vibrios, and particularly these other Vibrios, Volnificus and Parahemolyticus, a food pathogen that are being detected. And so we're in the process of doing that. And earlier, some years ago, we had done a study that was funded by NOAA that brought together a team in Oregon, in Louisiana, and in Maryland on Vibrios, and those data have been published in a book on Vibrios in the environment.
Very good. Well, let me come back to the question I posed earlier. So, as you know, the National Academies have been very active in this area of diversity, equity, inclusion. Almost every university has some program paying attention to this. And I wonder whether you think, from your journey, have we got this right? Are we headed in the right direction?
It's really necessary to bring all the talent into capability and action. We're in a global competition, which I can say, you know, having looking back over 50 years of science, there was a time 50 years ago when it was necessary to help the developing nations and work with our colleagues in Europe and in Latin America. And now science is global. And it really is important because we now are in a competitive world. And that's a good thing. But it means that we as a nation need to have all of the brains. You know, I'll put it this way. If you're a general of an army fighting a major battle, you're not going to just use half your army. And that's what we're doing in the science competition if we don't bring in the women and the other underrepresented minorities, because the brains are important. And there's another reason, and that is when you bring in a broader perspective, for example, in industry, if you have a board that has more than one underrepresented individual, if you have 3 or 4 women, half women, several African Americans, Hispanic, you make more money and that's been proven. I'm not making it up. Studies have been done because you're bringing a greater perspective and a wider view on the issues that are being addressed. So I think it's frankly imperative that we bring all of the brains for bringing back the chip industry, the manufacturing, for being able to stay competitive in space and to keep our health developments at the forefront. We just need everybody. We need all the brains. And so the argument to me is powerful. And the way to do it, I think there are many ways. I think we can spend a whole nother hour on that. But fundamentally is understanding really that we really need all the brains, is what I think the message is. Does that answer the question, Bill? Or
Well, yeah, I know when I started paying much more attention to healthcare 20 years ago now, it turned out the majority of my PhD students were female once I moved into that arena.
right,
And so some of it is not just good intentions, some is the way we package, the STEM disciplines in our research studies and stuff, not just having good intentions. My mother used to say that the road to hell is paved with good intentions. And so I think we need programs and efforts to really make a difference.
well, I think that's absolutely true. And I think an important part of it is that I think we need social structure like childcare. We need to provide opportunities, as I attempted to do and successfully at NSF. I started a program called Advance, which actually is open to men and women, but it's to provide opportunity at the university and individual unit level, with the responsibility being the senior officer, not putting the responsibility on the department chair, but rather on the president and the vice president for academic affairs and research, presidents for academic affairs and research, for the advancement and success of the program and the funding that NSF provides. And so putting the responsibility at the top and providing the opportunities in the entering level has proven quite successful. We did an analysis last year of the success of the advanced program, and we can account for about 9,000 women professors as a result of the advanced program funding. So that's one way that has proven successful.
Yeah,
But it's childcare that's critical, and COVID really wreaked havoc on young scientists.
Yeah, that's this has been really fascinating. I have to say it's at two levels. One is the level of what you've accomplished technically relative to COVID and cholera. The other is just your story of how you got there. Really fascinating. We appreciate your time, and I'm sure we've got a lot of people that are very motivated by what you've had to contribute today. Thank you very much, Rita.
Thank you for inviting me. It's been a genuine pleasure. Thanks, Mimi, too. So
Thank
Bill,
you so much
thanks
for
a
your
lot.
time.
Yeah.
Bye-bye.
Bye-bye.
And with that, we close out the 2022 SERC Talk series, and we'll be distributing this information as well as the recording and slide deck. The link to Rita's book will be available on the SERC website as well. And thank you all for joining us. Thanks for your time. Take care.
Yep.